Today, we are going to talk about the viral DNA and RNA replications and how they are distinct from one another. Viral replication is the formation of biological viruses in the process of infecting the target host cell. This is since viruses are obligate intracellular organisms, they are generally inactive outside a specific host. In the perspective of a certain virus, viral replication is done to allow the virus's production and survival on the host cell. Viruses can do this by generating copies of its genome and package into viruses that will eventually infect a new host. The pathway of replication is dependent on the viral components of the microorganisms, more specifically, their nucleic core. Viruses can either have a DNA or RNA as its genome, but not both. In the replication process of DNA viruses, only the positive strand is used while the negative strand is ignored. The positive strand uses the enzyme DNA-dependent RNA polymerase to synthesize mRNA. This mRNA produced would then undergo the translation process to produce the structural proteins and enzymes of the virus. DNA viruses usually replicate in the nucleus of the host cell. However, pox viruses are unique among DNA viruses because they carry out replication in the cytoplasm. These unique viruses produce cytoplasmic mini-nuclei, which are distinct sites that are surrounded by membranes derived from the rough endoplasmic reticulum, or the RER, which supports its replication process. For the viral replication of RNA viruses, the positive and the negative strands have to be considered, and this will dictate the pathway for its replication. In the positive strand or sense strand, the replication occurs in the cytoplasm. Therefore, there is no need for transcription to take place, as the RNA virus can function as the mRNA itself. Note that most RNA viruses, but not all, do their replication using the positive strand. The RNA replication of the negative strand or the antisense strand takes place in the cytoplasm as well. A host-provided enzyme called RNA-dependent RNA polymerase catalyzes replication to convert it to positive or mRNA. Translation then proceeds to use this information contained in the mRNA to produce the proteins needed by the virus itself. There is an exception to one type of RNA virus, and this is the retroviridae. It is due to the nature of the virus to replicate inside the nucleus, even if it contains RNA as its genomic code.